Excellent. So welcome back, everybody. Mid-morning, late morning session. So we have Hadley Muller from the Centre for Sustainability using stable isotope analysis to understand the diet of feral cats on the Chatham Islands. Really looking forward to this talk. I'm a materials scientist, so it's got my attention. Thank you. Over to Hadley.
Sweet. So yeah, I was doing stable isotope analysis on some feral cats over the summer, or still am. And I mean, it's a little bit of a change away from some of the social science projects out of the CSAFE. So it's a bit of ecology, bit of stable isotope chemistry. And I was supervised by Silawiki, and I also worked with Sarah Burry and had the opportunity to head up to Wellington to the NIWA labs. So feral cats in New Zealand, they are a really big problem. And they were kind of introduced to New Zealand from the get-go by European settlers, initially by James Cook, and then kind of continued to be introduced as companions, and then also as a way to control plagues of rats, mice, and rabbits. But they don't need us to survive, and they've been really successful and are an important invasive predator, and they've contributed to the decline of a lot of native animals kind of around the country. And nowadays, they tend to rely less on our native birds, and on the mainland, they have a really highly specialized diet. So this means that they tend to focus on maybe a single prey item. And this becomes a problem in island ecosystems. So for example, on the Chatham Islands, which is where our study is based. So in these island systems, there tends to be less diverse ecosystems. So they might not have as many native animals in the first place because they're so isolated, but also there are less introduced animals. So say on the Chatham Islands, they don't have any introduced rabbits or stoats or ferrets or anything. So what they tend to do is the feral cats tend to rely more on the native animals that are there. And this is a really big issue because island endemic species are often really long-lived. They might take many, many years to start to reproduce, and they might reduce really, really slowly and spend a lot of time on the ground, and they don't have any appropriate response to mammal predators because they're adapted to really only dealing with other predation from other birds. And so Chatham Islands has not escaped the problems of steep declines in endemic fauna across kind of these isolated island systems. And one of the most important species on the Chatham Islands is this bird, the Chatham Island taiko. So this is a ground nesting mutton bird. And it was initially discovered in the 1800s by a research vessel in the Pacific Ocean. And then it was kind of lost to science, or Western science, I should say. And it was rediscovered in the Tuku Nature Reserve in 1978 on the Chatham Islands. And there were fewer than 150 birds. So it's thought that probably habitat change would have reduced their numbers initially, but then after that, cats are believed to have been the primary threat. And we know that their decline has been very, very steep. And at the turn of the 20th century, the Moriori on the Chatham Islands were still collecting hundreds of birds every year. So the main issue is that these recently fledged birds, they spend their time on the ground and they don't even make it to the ocean as a seabird. They get eaten by cats before they even get there. So there's no hope for them reproducing. So that kind of brings the study to our aim is to understand the feral cat diet, and we are specifically interested in its variation over time. So the Chatham Island taiko is only breeding from September to April. So we want to see if there's any variations in the diet over time that might indicate that during the breeding season, they start to rely on these recently fledged chicks. So our study is based in the Tuku Nature Reserve, which is, as I kind of mentioned before, the only naturally persisting colony of the Chatham Island taiko. And there's been really extensive feral cat trapping there since 1987. So there's 20 k's of trap lines, which you can kind of see. And there's also extensive bait stations for rats and mice. And so our study, we're looking at 21 feral cats. They were trapped in 2017. So I know that stable isotope analysis is not a just a different form of an element that has a different mass. So I'm sure some people may have heard of kind of carbon-14, which is it's a radioactive form of carbon, but there are other forms of carbon, carbon-13 or carbon-12, and these are stable, and they exist in a natural ratio in natural systems. And we can use this ratio to take some conclusions about these complex natural ecosystems, because the different isotopes will move through these systems in a very predictable way. And in stable isotope analysis, we generally use C delta values, which is just means that there is more of the heavy isotope. So in our study, we're specifically interested in nitrogen and in carbon. So nitrogen is really, really useful because it can tell us about a feral cat's diet or trophic level, because there, as you can see in the figure down the bottom, there's a measurable and very consistent difference between the trophic levels. So there's about 2 to 3% difference between a predator and its prey. And then carbon is not as useful in our study, but specifically what it can do is it can tell us the origin of the carbon, which is a bit airy-fairy, but mainly looking at whether the carbon has come from a marine system, or marine carbon would be from the Chatham Island taiko. So it should be pretty clear if these cats are predating on these endangered seabirds. So we are looking at 3 tissues from these feral cats. So we have claws, we have fur, and we have bone collagen. And so these tissues represent different time periods of the cat's growth. And so we can then use the different time periods to look at variation over time. And this is kind of stable isotope analysis' big advantage over just cutting open these dead cats and seeing what's in their stomach. One, some things digest much easier than others. So if you cut open a cat's stomach, you might see that it's eaten a rat. But if it was eating insects, you might not know because they will have just digested away. And also by looking at the entire life. So we kind of, oh no, I've kept that in, that's okay. I kind of only have preliminary results at the moment. I have kind of just recently gotten back from a trip to Wellington up at Niwa. And so we're still waiting for a few of the results to come back. And we're hoping to do more analysis on this data. But at the moment, I can show you some interesting things. So this is kind of looking at the cat tissues here in the middle in the colors. And then all of these little other dots are all of the possible prey items that exist in this ecosystem. And so on the x-axis, we've got the carbon. So again, we might expect terrestrial organisms to be down this end, and marine organisms to be up by where the taiko and the fish are. And then on the y-axis, which is the one that we're most interested in, we would expect the plants to be down the bottom, and then the insects that eat the plants, and then the rats that eat the insects, and so on and so forth. And so you can see that in the middle here, the cats are all grouped kind of just above where the rats are sitting. So we would expect that the cats are have a higher delta nitrogen value than whatever their prey species is. So it's kind of looking like they are primarily subsisting on rats in the system, which is actually really great news because this is a system where the cats are really heavily controlled. And so it seems like, or it suggests that the conservation that's ongoing is keeping these cats at such a low level that they can subsist entirely on rats without reaching really high levels of population and starting to spill over into predating on these other native animals. And this graph already gives you an indication that there doesn't seem to be a lot of variation over time in their diet. And this graph here is quantifying the variation. So effectively, it's just showing how much variation is there in the diet over the different time periods. So remember, the claw is the smallest time period, and then the fur, and then the collagen is its whole lifetime. And so it's kind of giving us an estimate of the variation in its diet and of change over time. Certainly, over its lifetime, there's slightly more variation. And this is not super surprising. Some studies have just suggested that maybe in early spring or summer, before the numbers of introduced rodents start to pick up, that cats might rely slightly more heavily on birds or insects. But this is something that we still have to find out. So like I said before, we still have a little bit more work to do on this project. And we're hoping to be able to estimate the diet proportions, which is really that's really the killer. But the project overall should have some really cool conservation implications for the Chatham Islands. So it kind of clarifies the effectiveness of the current management strategy on the islands. So the Chatham Islands is quite a big island, and it would be really hard to run an eradication campaign, which has been done on other really small breeding islands. So it's really important to understand whether suppressing these cat levels, is that actually an effective use of money? Or would it be more effective to build a predator-proof fence and just call it quits in these unprotected areas? And also, this will be the first study to look at stabilisative analysis of cats in New Zealand, and certainly the first one to look at cat diet on the Chatham Islands. So we can say something important about the ecology of cats in these simple ecosystems that don't have as many players as the mainland. Yeah, so I'd just like to say thanks again to the Centre for Sustainability, the Stable Isotope Lab up in Wellington, which was really exciting to get the opportunity to visit that. And Nikki, one of the lab techs in the Department of Zoology, was really helpful during the project as well.
Thank you, Hadley. Well done. Very much enjoyed that talk. I liked how you're essentially seeing back in time and getting a really good idea of what those cats are predating on. You're frozen, so we'll just know that you're back. That's good. Any thoughts about, often I work in the ag sector, and you hear comments sometimes that cats, we don't want to get rid of feral cats. They do a good thing, suppressing rats and mice and things like that. And what you've just presented might sort of support that argument. Your thoughts on that?
It kind of, I guess it's hard to, if there are only 150 of these endangered seabirds living in this nature reserve, then the chance of us sampling a cat that has predated on these Chatham Island taiko is really unlikely. But it doesn't disprove that they're doing that. Like, it would only, and we know from the species that cats are still having a really negative impact. That Chatham Island Kereru, after cat in the first 3, it tripled its population. So they're definitely having an impact. But yes, I agree. Feral cats are kind of hard because they're obviously domestic animals as well, and people have them as pets. So it's hard to reconcile kind of a predator control in some settings. But yeah, I guess it has to be done.
Thank you. You were breaking up there, but I got the gist of your answer, and it's really informative. And so we'll use that then as a way to let you off the hook, since you're breaking up. No more questions. Well done, and thank you.
Thanks, Hadley.
We are right on time. Thank you. Next up, we have Charlie Cox. Is Charlie on board? Brilliant. Charlie, do you need me to pop up the slides, or you've got them there?
No, I've got them here.
Brilliant. Thank you. And so Charlie Cox from the Centre for Sustainability will be talking about organic carbon loss from earthquake-induced landslides, in Fiordland. Over to you.
Cool. So can you see that?
Yes, we can. Thank you.
Cool. All right. So hi, I'm Charlie, and I'm gonna introduce you to Fiordland earthquake-induced landslides. And so do you think that these landslides could play a big enough role to have an impact on New Zealand's carbon budget? Well, these are some examples of the landslides from the 2003 magnitude 7.2 Fiordland earthquake. Some debris flows, regolith failures, and wedge failures. And notice the scale in the middle picture. It's quite hard to see, but you can notice the snow line is less than, or the landslides reach more than halfway up to the crown of the landslide to the snow line. So they're quite big landslides. And in New Zealand, we account for all of our carbon emissions across all sectors of society. And we also account for our carbon sequestration in the land use change, land use and forestry sectors. So in 2020, we emitted 9.2 megatons of carbon, so that's 9.2 million tons of carbon, and we sequestered 6.3 million tons of carbon. And in New Zealand, we count any deforestation as a total loss of carbon. So none of it is counted as storage. It's all just counted as a loss as soon as it's deforested. And so this study, we mapped two earthquake events: the 2003 magnitude 7.2 Fiordland earthquake and the 2009 magnitude 7.8 earthquake. And for the 2003 earthquake, we mapped 1,852 landslides, which had previously been mapped as 433 landslides. And for the 2009 one, we mapped 313, which had previously been mapped as 187. And over 40% of these landslides were within 20 meters of the rivers, streams, and fjord network. So they're subject to transportation in the fluvial and marine environment. So as an example of the mapping, on the right is an image of some of the mapping process, and you can see the source areas in dark blue and the debris trails in light blue. And for these landslides, huge amounts of vegetation and soil was entrained and moved laterally. And in Fiordland, we have a very high carbon sequestration in the vegetation, but actually equally as important, and in some places more, we have organic carbon sequestration in the soil. So some people might think when you're accounting for carbon loss in landslides, you'd only account for vegetation, but in places like Fiordland with very densely vegetated hill slopes, that it's as important to look at the soil as well. So for vegetation carbon, there's a paper from on carbon amounts in different vegetations in New Zealand, and I combined that with the land cover database in New Zealand from Landcare Research, and with vegetation types, I got carbon density for a series of land cover types in Fiordland, And for soil carbon, I have a 1 kilometer resolution raster, which was a concentration percent of carbon from 0 to 30 centimeters of soil, and that was sourced from Pierre Rudier and at Manuka Whenua Landcare Research. And that's a national database. And yeah, is quite a newly developed database. It's just pretty much being used just in this area at this stage, but it could definitely be implemented along with the land cover database in other areas of landsliding, such as Kaikoura or the North Island, from some of the cyclones we've had recently. So as a recap, in 2020 we had 9.2 million tons of carbon emitted and 6.3 million tons of carbon sequestered. But in the 2003 and 2009 earthquakes, 2.2 million tons of carbon was transported by landslide. So that's over a third of a year's worth of carbon sequestration from 2 earthquakes. And that carbon will either biodegrade and create greenhouse gases, or it'll be stored in the fjord sediments. But it's kind of unknown and a bit controversial how much carbon is actually stored in the fjords, because we have quite small fjords. So a lot of it, if it ends up in the fjords, will be transported offshore and stored in the marine sediments, or biodegrade in the marine environment. And this graph, it is from a satellite-based, remotely sensed data set on global forest change. And it spans from 2001 to 2022 and shows annual loss of forest. So you can see the 2003 and 2009 earthquake events on the graph in blue and red, but all the other years could be understood as rainfall-induced landslides, and they total to the same, if not more, area than the earthquake-induced landslides. So especially in places like Fiordland, where you have really high rainfall, it's really important to think about rainfall-induced landslides as well as earthquake ones. And the return period for earthquakes of a big enough magnitude to have these landslide events is about 10 years. So yeah, in terms of rejuvenation of bush, that's on the left is a map that shows some of the landslides from the 2003 earthquake, and on the right is a photo taken 20 years after the landslide. So Fiordland is quite steep and has really shallow soil on top of rock, and when you lose the soil off the hill slope, it inhibits the rejuvenation of the bush, and also the soil takes a long time to recover. So this is 20 years worth of rejuvenation, and you can see the contrast between the slopes that haven't had landslides recently and the landslide scar itself. So a lot of people would think landsliding would kind of create places for new bush to grow, and young bush sequesters a lot of carbon, but the time scale that this is happening kind of makes it a lot less relevant than the loss of carbon from the landslides. So in a summary, there's over 2,000 landslides mapped from the 2003 and 2009 Fiordland earthquakes. There's 2.2 million tons of carbon lost. And in 2020, New Zealand only sequestered 6.3 million tons of carbon. Satellite observations show that the annual loss from rainfall is as much, if not more, than earthquake-induced landslides. And in Fiordland, the rejuvenation of native forests is slow. So in conclusion, the landslides are extremely important when accounting for New Zealand's carbon budget. If I'll just flick back to this slide here, and I just forgot to say that it's important to think that you notice the magnitude of the earthquake versus the magnitude and distribution of landsliding, and actually it's not the size of the earthquake that controls the magnitude and distribution of landslide, but it's more the topography and the ground acceleration. So yeah, it sort of isn't so important on the size of the earthquake, on how much vegetation is lost, but it's more so just dependent on where the earthquake is and the ground shaking as well. So yeah, cool. I think that's me. I'll just work out.
Oh, sorry. Well done, Charlie. That was great. This is Caroline here in the audience. Fantastic. I'm very proud of the work you've done over the summer. Just could you comment on the implications of this study for future earthquake hazards that we know exist? And this project kind of came from an interest of mine on the Alpine Fault, and we know that the Alpine Fault could produce a magnitude 8+ earthquake in the next 50 years. There's a very high likelihood of that. So what could you say about your study that might have implications for significant landslides following an Alpine Fault earthquake?
Yeah, so an option for continuing looking at this, there's quite a lot of data on the Inangahu, Murchison and Kaikoura earthquake events. So we could look at a similar process for those and sort of work out kind of a model of what's happening for each earthquake. But in terms of an Alpine Fault earthquake, the same could be done with some of the previously mapped landslides from the Alpine Fault stuff. But the way that the vegetation varies across, I guess, the Southern Alps and across kind of traversing the South Island, there's so much variation in vegetation type that it's a lot more complex than a place like Fiordland that has quite homogeneous vegetation.
Yeah. And the other sub question was around the regeneration part of your study, where after 20 years, it didn't look like a whole lot of regrowth had taken place. What role do the browsers like deer play in countering some of that regrowth?
They probably play quite a big role, but a lot of the slopes that have failed are quite a high angle, and it's probably I've seen a lot of the scars from in the water, and a lot of them are places where you wouldn't expect deer and stuff to go. Many of the landslides that we mapped are pretty much vertical, if not overhanging in places. But I guess there's quite a lack of good data in digital elevation models in Fiordland, so that would be a really important thing for looking at that, would be having a new digital elevation model for Fiordland. Yeah.
Thank you. Well done. Excellent. I'm a West Coaster, so that definitely got my attention. And I was born 9 months to the day after the Inangahoa earthquake, so I think the earth did shake at my conception. But anyway, that's oversharing. So we're on to our next talk.
That's a great claim to fame, by the way. I'm gonna use that.
Oh, the name amongst my aunts and uncles referring to me as the Inangahoa kid.
Okay.
Anyway, so we're on to our third talk of the session. This is a joint presentation with one student remoting in, Georgie. Brilliant, you're on board. And Anna and Irris. We will get you up here.
Hello, can everyone hear me all right?
We can.
Yeah.
Brilliant.
Okay, perfect.
Got that, we got that, we got that. So Hannah, Irris and Georgie will be talking about kelp biostimulants and microbial bioformulations. They are students from the Agricultural Innovation Program.
Sweet. Good morning, everyone. I'm Hannah and I'm a fourth year micro and ag innovation student.
Kia ora and good morning, everybody. My name is Irris Etches, and I'm a second year Ag Innovation and Computer Science student.
And I'm Georgie, and I'm a third year Ag Innovation and Environmental Management student.
All right, I will be starting us off with our presentation. So firstly, the first point is, what are biostimulants? We've heard a lot about them. I'm not sure if you've heard of them in the news recently or in the ag space, but I'll give you a little bit of a background on it, why we use kelp, and a little bit about kelp blue. So a biostimulant is any substance applied to plants with the aim to enhance nutrient efficiency, abiotic stress tolerance, or crop quality traits, regardless of its nutrient content. Biostimulants stimulate the natural process of the plant by enhancing its natural nutrient uptake and tolerance to abiotic stresses, which improves the quality of the crop. Biostimulants differ from fertilizers because instead of adding nutrients to the soil, it stimulates the plant's nutrition processes independently of the product's nutrient content. And adding a biostimulant promotes the plant growth, and it increases the tolerance against abiotic stress, as I've been mentioning. So for example, that includes drought, salinity, and rain. StimPlus is an example of a kelp-based biostimulant that was used in my lab scale trial. My second point is on why we use kelp. Population growth and strained resources have pushed farming industries to use one crop in different ways. There is a growing interest in kelp from several sectors, with a particular focus on their potential as a source of nutritious food to feed the growing human population and the ecosystem services they provide, particularly in reducing greenhouse gases. As we know, climate change is an uncontrolled factor in farming, so there is a need to find an alternative, cost-friendly, environmentally friendly plant stimulant. Macroalgae works in the favor of farmers by containing properties needed to tolerate abiotic stress. And as you can see in the last point, brown algae, it contains all of these valuable extracts and like fluorotannins, fucoidanes, and there are other active compounds in it. An important point of seaweed is that it can perform carbon sequestration while providing industries with food, medicine, supplements, and agricultural products. This approach to farming aligns with sustainable agriculture. And my third point is on kelp blue, just a little bit about them. It is a biotech company that produces kelp-based biostimulants. Giant kelp is the seaweed used in their liquid biostimulant products, and the methods employed during production are sustainable in the sense that every component of the harvest material is used. I will now pass this over to Georgie.
Hi there. So quickly on the adoption of biostimulants in agriculture was definitely unclear over the last through the 70s and things. So now that the frameworks and definitions and there's more biotechnology in creating biostimulants, farmers can now adopt this more easier. So with the push towards sustainable agriculture and stricter chemical regulations, biostimulants are becoming necessary for alternatives meeting these growing food demands. Additionally, the further understanding in them has been developed successfully. So from farm and an increased yield and ROI has been found, as well as resilience to climate stress and soil generation. Next slide, thank you. And this is where the aim of my project comes in. So our aim was to test the performance of kelp blue seaweed based biostimulant across pastoral, horticultural and cropping sites. This is an important trial because it not only shows biostimulants potential, but shows how they interact on two working farms, and then using that information that we've learned from the last 50 years of biostimulants to make more informed decisions on how to apply them to farms so they can be widely adopted. So the key metrics for the field trial was to measure a range of critical variables to assess the biostimulants impact, which included pasture quality, yield, protein content, water soluble carbohydrates, and a whole lot of other things. And then these metrics are used to understand the biostimulants efficacy and enhancing the soil health and crop productivity. So we've had a bit of a larger timeline. So this will go out until May. And we've started with the baseline soil testing, and then we've applied the biostimulant and a fertilizer. We've also had pesticide application, and then we've harvested each month as well. So each phase is carefully documented with specific dates and locations across the two test sites. One of the test sites is a conventional property, where the other one is a more regenerative property. So we're looking about how those make an impact on the data as well. In this picture, you can see we've done the field tests. The bottom middle is the established after the seed had been directly drilled, and then the top one is that growing after a little bit. Next slide, thank you. This is the experimental setup showing the placements of the grids, and then the different applications and where they're going. At the right picture is a application plan for one of the sites, the conventional site, which shows all the different chemicals, fertilizers, pesticides, and the kale getting drilled over the period, which is really interesting to look at because it comes to about $4,000 worth of fertilizer and $1,300 worth of chemicals on this 10 hectare crop. So we're looking at how much is being applied to that area versus the regenerative area, which has only just had the seed drilled and glyphosate at the start. Next slide, thank you. So in this thing, you can see this is the regenerative site that has the mixed pasture species. This image kind of suggests that the combination of the biostimulants and the fertilizer is producing a more resilient crop, as the evidence appears that like the biostim and FIT combined section controlled compared to just biostim or control or fit sections is doing a lot better. And I think that's important to recognise that while the fertilisers have traditionally been used to increase the crop yields, their singular application only focuses on the quantity of the produce rather than the overall health and resilience of the plants. So the addition of the biostimulant appears to be supporting the plants in a way that not only grows the plant, but also inherits the defense mechanisms against the stress factors like disease, pests, and other conditions. So we're starting to see some good results already. And I think the integrated use of the biostimulant and the fertilizer together is not just about boosting the yield, but like fostering a more kind of robust crop. And this is interesting to look at from a large scale, as just applying the biostimulant would not be effective possibly at this stage. And if you were going to put this in a conventional environment, you could probably look at reducing some of your fertilizer, but pairing it with the biostimulant. Next, thank you. Oh, and I'm passing back to Irris.
Thank you. This was the lab scale trial that we ran here at the university. And from the pictures on the left-hand side, you can see that the kale grown in where the blue arrows are pointing, they were grown from soil from a conventional farm. The leaves are much bigger compared to the leaves from Lake Howea Station. And from this observation, we can see that this is likely due to the excess nutrients that conventional farming is known for, or the methods adopted on the farm are known for. Lake Howea Station is a regen farm. So that is a comparison that is good to note for future reference. And on the right-hand side is ryegrass that we grew, and it's currently growing, and it's an ongoing process. It's an ongoing treatment, still undergoing treatment. And the 3 different health biostimulants that we used were from a local Dunedin company, an international company, and an iwi-owned farm in the Sounds. They were treated with kelp biostimulants at different points, and the results will be written in my report. And I will pass it on to Hannah.
Sweet. So again, I'm Hannah, and my project was focused on the use of microbial bioformulations for seed coatings to enhance germination rates and thus increase crop productivity. My study addresses a challenge of the maintenance of microbial viability during the seed coating process and subsequent storage. Microbial bioformulations offer a promising avenue to enhance the sustainability of agricultural practices. So firstly, I'll introduce you to the plant-microbe interaction. So a plant-microbe interaction is the relationship between plants and various microorganisms, including bacteria, fungi, and viruses, that can either be beneficial or harmful to plants. These interactions play crucial roles in plant growth, development, health, and overall ecosystem function. A symbiotic plant-microbe interaction is a mutually beneficial interaction between plants and certain microorganisms, where typically the plant exchanges organic compounds for essential nutrients such as phosphorus and nitrogen. By incorporating beneficial microorganisms into seed coating, we aim to establish a symbiotic relationship between these microbes and the germinating seeds, promoting plant growth and offering a protection against pathogens. For the study, we use Stenotrophomonas rhizophila, a bacteria, as it's known for its positive impact on plant health, and we used ryegrass as our plant. So the challenge that is faced by attempting to promote the symbiotic relationship through seed coatings is ensuring the viability of these microorganisms throughout the seed coating process and storage. Environmental factors such as temperature, humidity, exposure to oxygen, and many other factors can significantly impact the microbial survival, leading to a reduction in the efficacy of the formulation. So to address this challenge, my research project was to explore the potential of two natural polysaccharides, xanthan and guar gum, as thickening agents in a gel to improve microbial viability. These polysaccharides are known for their ability to form stable gels, making them ideal candidates for enhancing the survival of microorganisms. So xanthan is an exopolysaccharide produced by the bacterium Xanthomonas campestris, and guar gum is derived from guar beans. So it was hypothesized that incorporating these gels into the seed coating will create a protective matrix around the microorganisms, mimicking a biofilm structure, safeguarding them from environmental stresses and in turn enhance their survival. Our methodology involved coating ryegrass seeds with either xanthan or guar gels with the inoculant. We also used a positive and negative control. These coated seeds then underwent simulated storage conditions, and we assessed microbial viability periodically. Additionally, we monitored germination rates to evaluate the impact on these gels on the overall success of seed germination. So to evaluate the efficiency of our formulations, we estimated the number of microbial cells on each treatment group through serial dilutions and colony counts. So the more microbes that get onto the seeds and that are alive, the better the formulation. So these were a few of our results. This is the estimated number of colonies coded on the seeds for each treatment. We used colony forming units per gram to indicate microbial presence in each sample. These results show that xanthan gum treated seeds had a high microbial presence, and both gels had higher microbial load than the controls. So it kind of just proved that it worked. So this was our glasshouse trial. We planted 5 pots of 10 seeds for each treatment, as shown. You can't really see much of a difference in the photo between treatments, but in our dry matter results, shown here, you can see that guar and xanthan coated seeds had the highest dry matter compared to the controls. And our seed storage experiment is still ongoing, so we don't have any significant results yet. The outcomes of this research hold the potential to contribute significantly to the development of robust and effective bioformulations for seed coating. Xanthan is likely to be further experimented with in the future as it can be used in organic farming. And by understanding the effectiveness of gums and increasing microbial viability, we aim to inform agricultural practices, fostering sustainable approaches to enhance crop productivity. And I pass on to Georgie for our concluding words.
Saying she can't say thank you as she's in the office. Can you please do it?
Okay. So lastly, we just wanted to thank our sponsors for giving us some funding to be able to do this, and thank you, Craig, for the opportunity to be able to do this. We've all learnt a lot, so thank you.
Thank you, guys.
Well done.
And thank you, Georgie, for being chairing this session better than me. We can go over time because it's the last presentation in this session, but well done. Question? Yes, please.
If you want to.
Yeah.
Okay. A quick question for Georgie. With you gave us a sort of a relative cost for the fertilizer across that field site, and it was quite a lot of money. You know, it sounded like a lot. How would it compare using a kelp based product? Would it be a lot cheaper? So that's the first part of the question. And the second part is, if these kelp products are seen as a fantastic way to, you know, support regen and get the stuff cranking along, is it a sustainable industry? Is there enough of this stuff for us to use and apply more widely? Sorry, two comments.
Yeah, perfect. Well, that's the exciting thing about the fertilizer and I think there's a lot of space for it already on regenerative properties, but I think it's more working out how to integrate it into a conventional property so they can cut the costs of a fertilizer and then apply the biosimilar together. I think that's the most valuable way that's going to have a impact on the wider farming industry. And I do think that there would be enough for wide spread adoption. They just need to continue figuring out their systems. Yeah, and
What
that's
about-
what they're established in Namibia, Alaska, and New Zealand at the moment. And once they've kind of got all their protocol underway, they will be able to mass produce enough.
Oh, that's great. And what about the
It's
cost?
fine.
Will it be quite a lot cheaper than regular ferts, or are we looking at?
I think it will. I think it's just quickly important to like acknowledge you can't replace fertilizer with it. It's something that would be used to pair, but it would definitely reduce the cost of fertilizer, and I think it would be quite price competitive to be able to implement that into a conventional farm setting, if that kind of makes sense.
Thanks, Georgie.
And
So,
yeah.
Awesome.
Yeah, both biostimulant kelp-based ones we've been looking at farmed kelp,
Yep.
which is a big plus.
Yeah.
And not interfering with natural ecosystems.
Yeah, that's great. Fantastic.
Any questions from online? Well, thank you. Thank you, guys. Thank you for a great presentation.
It's lunchtime-ish now,
Lunchtime-ish.
but not quite yet, I think. But if we're not back to 1:15, I think is the starting up again. Yeah. Great, excellent. That's well done.